Hello, my name is Juan Flores, and I will be sharing with you about my project where I compare the volatile compositions and chemical richness of the four fungal species that are listed below. So the first one is Leptographium longiclavatum, the second is Ophiostoma montium, the third is Grossmania clavigera, and lastly the fourth Penicillium exansum. And I compared the compounds that these four fungal species emitted, shown in two studies, and however, my results showed that these compounds cannot be compared to one another due to both studies being very different in methods and standardizations, where no conclusion can be made. And so what I did was I created a principal component analysis for the volatile composition of four fungal species, and I did this by comparing the concentration of their volatile compositions for the four fungal species that are listed here in this figure. And as you can see, P. exansum has the largest difference from the other three, and these three a belong to paper 2, and then P. exansum is from paper 1. But as you can see, the P. exansum has the largest difference in PC1 and PC2 axes, and this essentially means it's the most different in the concentrations of the volatiles that these fungi emit. However, based on this figure, we cannot make a conclusion due to the bias that will be shown in the next slide, but the bias in the standardizations and methods that both these papers use make a conclusion not available. So I also counted the compounds that each of the four fungal species emitted, and as you can see, P. exansum had the largest with 91 volatiles measured, which is from paper 1, and then the other three from study 2 were only measured for 9 headspace volatiles, and so G. clavigera emitted 6, L. montium emitted 5, L. longiclavatum emitted 8. As you can see, there's a large difference between study 1 and then study 2 in terms of the number of compounds, and this essentially is what created the bias of making P. exansum very different from the others and the principal component analysis, and therefore we could not make a conclusion. And you may wonder why this applies to the real world, and as most of us know, fungi causes crop loss because it contaminates crops as shown in the images here that makes them inedible. However, we also know that fungi emit volatiles, and as we learned in chemical ecology, these volatiles can be used for interactions and communications between fungi within each other. And so this is essentially what my study focused on. They wanted to analyze the interactions the similarities that the volatiles have to see if there's some hinting of some interactions that these fungi undergo within each other. And this can then further lead to further studies that analyze these interactions that would then lead to ways to prevent or limit the annual crop loss and help our agricultural systems. So to actually obtain this data, for paper 1, I pulled the concentrations of each compound within each class from the control group that is shown in this figure here, and the only calculations I did was I converted the concentration units micrograms per liters to nanograms per liters, and I used these concentrations to create my PCA, and then I also created a binary table to count the number of compounds measured in this study. And similarly, in paper 2, I pulled the concentrations for the 9 headspace volatiles measured for the 3 fungal species from this figure here. However, the concentrations were nanograms per millimeter squared, and I needed it to get to nanograms per liters. So I multiplied by the culture area that was provided, and that canceled out the area, and I just divided by the volume used to measure the volatiles, which was also given in the paper, and that got me to nanograms per liters. And similarly, I did the same. I used these for the PCA, and I also counted the number of compounds. And that concludes my presentation. Thank you.